so in this video we're going to see some of the techniques for staining. And before staining, you need to wear your personal protective equipment, or PPE, and includes wearing your lab coat, gloves, your glasses if you wear them. And then you have to clean here your area, your working area, with this disinfectant and with paper towels. And you will have as well in this area your stainings, your dyes, which in the case of one of the staining techniques that we will be doing is the grime stain, which includes crystal violet. It's the first stain that you add or dye. This is iodide, which is a mordant or a fixative agent. This is 95% alcohol. And then the counter stain, which is safranin. So they are applied in that order. And then we're going to do also simple staining, which includes methylene blue. And we're going to do something that's called negative stain, which is nigrosin. And you will have in a rack your cultures. So in this case, I'm going to use Micrococcus luteus, which is a small spherical-like bacteria. And then this is a broth, and this is a slant. So a slant is a solid media with a little bit of an incline into it. And then you can see the growth of the bacteria there. So you have to keep it in your rack, and anything that you have in your countertop, it shouldn't be close to the edge, otherwise it will fall. So those are general precautions. Now, here you will have your container with different items that you will need. So you will need a lighter for turning on the Bunsen burner. Then you will have your microscope slides. These ones are matches, you don't need them. You have some Sharpies, and then you have some clothespins so that you can manipulate your slides. And then you have scissors. You have some rulers and some tweezers. But for the stainings, you will use your marker, your microscope slides, and one of these. So then you will have everything, and also your squirt bottle so you can wash your slides when you're doing your techniques. And I'm going to grab a piece of paper towel to start cleaning this area. And the first thing that we're going to learn is how to make a smear. So what is a smear? So it's basically a thin film that you put into the glass slide, in the surface of the glass slide, and that film is going to be bacterial film so that you can work with your bacteria. Now, in the case of some stainings, you will be using bacteria that it was fixed and killed with the heat from the Bunsen burner, but in other cases, you won't do that. In which case? Well, in the case of the negative stain. So, in order to turn your Bunsen burner, what you will do is open up the gas valve, and then you can listen that the gas is coming out from there, and then you just strike your splitter into there, and you will have to take your glass slides by the edge, and they're supposed to be clean, and you will label them appropriately. So you use this third part and the top part to label whatever bacteria you are putting into there. So in this case it's Micrococcus luteus. So I'm going to just put capital M and then luteus with lowercase, and then today's date, and then your initials. So that's what you will do. And then depending on what you are planning to to do, but sometimes it is advisable to label the bottom of the slide with a circle so you can identify where you have put your bacteria. So I'm going to do that, or you can do a couple of lines. So I'm going to make this little circle, and it has to be on the bottom of the plate, not on the top. And then you will have as well these inoculating loops. So this is an inoculating loop. It has this metal wire, and then this is an inoculating needle. Now for the stainings, you will use this inoculating loop, and then you have to use the aseptic technique. So basically, this is the sterile area. The heat is killing all the microorganisms around, and then this wire, you have to put it in the center of the flame at a 45-degree angle, grabbing it like this, like if it was a pencil, with your index, your pollex, and your middle finger to make point of support. And then it has to kind of be like in this area of the handle, not too close to the edge, not too close to the part where you have the loop. So it has to be somewhere in here so you don't burn yourself as well. Put it in 45-degree angle, and notice how the wire is getting red hot, meaning that you are sterilizing this wire. So you withdraw the wire once it's red hot from the flame, let it cool. That's kind of one of the longest steps. Let it cool for, let's say, around a minute, and then from there you will grab your bacterial sample, and then you will start doing your smear. Now when you are doing smears from a liquid media, you don't need to previously add water as your manual indicates, and this is just because water acts as an agent to help the smear to become very thin. And if you add solid media, you have to have a drop of water into the area where you're going to set the smear, so you can have all this film very thin, and then you don't over having a lot of bacteria preventing the stain or the dye to penetrate into the bacterial cells. So first I'm going to make then this smear using this liquid media, which is a broth. And then the liquid media, when it's non-sterile, when it has bacteria, it looks like this, turbid in color or opaque. And then what you can do to suspend better the bacteria, because by gravity the bacteria tends to go to the bottom of the tube, as you can see there, you just have to kind of shake it a little bit, very careful to not spill, and then bring your rag close to this sterile area, and then you will open the cap or the tube, withdraw the cap next to the sterile area. You withdraw it like this, the cap, and then pass a couple of times the opening of the tube and also the cap like this. So this is to prevent any microorganisms from being on the tip of the tube. And then grabbing like in a 45-degree angle, you introduce your loop, and then grab a loop full of bacteria from there, put it in here in the glass slide like this, and then close your test tube by flaming it again, put it on the rack, and then you will grab your glass slide, and then in a circular manner work around the area where you mark below in the glass slide. So over there the film is very thin, the smear, that's how we call the film. Let it dry, put it in there, and then sterilize your loop. 45-degree angle, and then let it cool. While this one is cooling, you will check against the light your slide to see if it's completely dry. So you can see there the area that still is not dry. And then once it's dry, what you're going to do is going to pass really quickly this slide against the flame 2 to 3 times. So I'm going to put the loop here as I'm waiting for this to dry. And again, this is one of the things that takes a while. And then of course, the more that you spread the film, the less longer that it takes to dry. But for me, that's enough. It's almost dry. I can see it, and I will show you once it's dry a close-up of how it looks. So another thing that you can do meanwhile is grab it with clothespin like this, and let it stay until it's completely dry. And as this is happening, I'm going to label my other slide for the solid media, the slant. And at some point, that's something that you can learn as well. While you're waiting for some things, you can start doing other things. And again, it's the same bacteria, but now it's going to be in a solid media. Use Micrococcus luteus, put the date in the slide, and then your initials. And then again, label the bottom. In this case, because it's a solid media, what I'm going to do, instead of putting a circle there, I'm going to draw 2 lines in the bottom of the slide opposite to where you label your glass slide. So that's the bottom, this is the top. Why you do that? Well, because since you're going to be adding into some of these staining techniques, you're going to be adding some decolorizing agents on the top where you have the smear. If you add this, basically part of the Sharpie, part of the colorant of the Sharpie will go into the bacteria. So this is almost ready for being dry. And then this is how this looks. So now let me show you how a good smear looks like. So as you see, it's not very thick. Thickness can be seen when you see a white film. And this is kind of opaque, but not that much. So this is then a smear that is being dry, and you have to heat fix the bacteria. So you will attach the bacterial cells to the bottom of the slide or to the surface of the slide when you are heat fixing it, and then you kill them at the same time. So I'm going to do that really quick. 1, 2, 3, and that's it. And then you put it in here, and then you start your staining technique. But let's do first this other one for this solid media. So when you're doing that for the solid media, what you're going to do once you sterilize your loop are going to add a drop of water into the mid portion of your sterile loop, and then put it in there, in the area where you're going to make your smear. So let me show it to you. So that's the water. And now, since you added water there, with your loop, what you need to do is to sterilize again your loop, because you don't know if the water is sterile or not. And you don't necessarily, in essence, I mean, in practice, you don't necessarily need to use a sterile loop, but you don't know if whoever used that loop before sterilized it. So that's why you sterilize it before putting the water. And then you have to let it cool enough so when you are introducing the loop into the agar, when you're going to grab the bacteria out of the slant, the agar slant, then you don't kill the bacteria or you don't destroy the actual microorganisms and as well you don't damage the agar. And then when you are grabbing bacteria from your slant, you're going to follow the same angle. And usually what you can do is instead of grabbing it from the inside of the loop, you just touch with the outer edge of the loop the bacteria, the colonies, the yellow colonies that you see there, and then you put it in the water that you have in your slide. And it doesn't matter if it doesn't look to you that you have a lot of bacteria, but actually you do. But sometimes you can make a very thick smear, and that's not good. So again, you have your test tube, open it next to the sterile area, pass a couple of times the opening of the tube and also the cap, and then 45-degree angle, introduce your loop, touch the surface, withdraw the loop, close your test tube, put it in the rack, and then grab your bacterial sample, put it there in the middle, and start mixing it with the water in a circular motion to make a homogeneous dispersion or mixture. And then you start spreading this along the area that you marked, and then wait it to be dry. So I can see now, it's hard with the camera, but I can see the whitish color that is coming from all of these bacteria mixed in the water. So I have done that. I'm going to sterilize my loop, and you can see even the spattering of the bacteria there as it's being killed, and wait for this to dry. And I'm going to grab another clothespin. Oops, wrong one. And then put it in there. So with this, I'm going to do ram stain, and I'm going to prepare another tube for doing this methylene blue stain or simple staining. And then the other one that I'm going to do is for nigrosin stain or negative stain. So, and I'm going to label them just while I wait for that to happen, to dry. So okay, so that one, it's almost dry. So while it dries, what I'm going to do is to explain the principle of the simple staining. So simple staining, the dyes have a positive charge. And because you have a positive charge, the dye will go into the inside of the cells, which are negatively charged. And then you will stain the cytoplasm. And you use a single stain. Now, in negative stain, the charge of the dye is negative. So that's why it's called negative stain. So since you have negative charges in the cytoplasm and you have negative staining or negative dye, the dye will not penetrate the cell, so you stain the outside of the cell. So now this one is dry, and look at the whitish film that you see there. It's a little bit opaque, so that's how it's supposed to be. This one, I'm going to just hit fix like this, and then you will do the same for your negative stains. And I wanted to do this smear very quick for the simple stain and the negative stain with this liquid media, so I don't have to use the water. And then sometimes you don't want to work with that part that you put in the bottom, like in the case of the negative stains, just because all the slide is going to be covered with bacteria. Now, I think I said that you will heat fix the slide when you are doing the negative stain, but no, you don't do that with the negative stain. So I'm going to make the smear for the negative stain, and in that case I'm just going to let it dry, and I'm going to put it there in one edge. While I put the other drop in here for the simple stain. So I'm going to distribute then this for the simple stain, and I'm not using, as you can see in this case, any of the marking that I did for the other two. So it's going to dry there, and then I'm going to sterilize this loop again, and while this one dries, what I'm going to do is going to start doing this negative stain. So in negative stain, what you will do is add a drop just where you have added your bacteria without touching the bacteria. And then you have a very thick drop in there, and you have to homogenize these bacteria together with the negative stain, and you have to have it in the opposite end where you have your label. And of course you have to let this dry, cool before you mix them, otherwise you will destroy the bacteria. And again, in negative stain, you have negative charges, so the stain or the dye does not penetrate the bacterial cells, so you stain the outside, and then you do this mixture in there, and then you will sterilize your loop. And with another slide, a clean slide, you will put your slide almost in a 45-degree angle in this way, opposite to the end, like this, until by capillary action, and you kind of drop it a little bit till you grab on the edges, as you can see there, the stain. And by capillary action, you will grab your dye in between the two slides, your stain, and then you will drag it like this a little bit towards the edge, and then on the opposite side like this. And don't let it fall, otherwise you will have your slide all stuck with this other one, and then you won't be able to see anything. So this is how this type of staining looks like. And then I'm going to put it for now here in the countertops. And you're supposed to have as well a beaker with disinfectant, which you will have. Now this one is dry. So what I'm going to do, this is for the simple stain. And I know because I didn't put this circle or these lines in the bottom. So for this one, I'm going to heat fix 3 times, pass it in there. And in this, you will use just methylene blue for 1 minute, and then you rinse with water. So this is the first one that I'm going to do. So I'm going to add in the area where I had this staining, or sorry, this smear, the methylene blue. Like 5 to 6 drops, and then count the time for 1 minute. And there's a clock there in the back of the room that I'm going to be using. Okay, so after that minute, you just check off the excess of dye, and then gently you squirt water into the surface of the slide. And then you can put it like this, and then it will rinse and dry. Now for the grime, you have again the 4 sets of reagents. Crystal violet, which is the primary stain. Then iodide, which is a fixative agent. And then you have the colorizing agent, which is alcohol, 95%. And then you have safranin, which is the secondary stain or the counter stain. So it will be 1 minute, 1 minute, 10 to 20 seconds, and 1 minute. So I'm going to add crystal violet, like 2 drops to each slide. And if you see in your slide that you are still not covering part of your smear, what you can do is just move it a little bit. Like in this case, I have more smear than that. So I'm going to move it a little bit to cover the entire smear. And yeah, you can start thinking about, well, the time is going to be not the same for the edges that I'm covering. Yeah, of course, but at least you know that in the center you have that. Otherwise, you can add more drops until you cover in the entire smear. It doesn't matter, and you can have it all at the same time. Okay, so after that minute, you rinse it, and then you do it like this, putting your slide sideways. And make sure that you don't have any dye left. you add your fixative agent. Again, 1 minute. And while that minute happens, you can start also shaking the excess off from your slides. There is one way you can use to let it dry much faster. It's by using paper, bilirubin paper, in which you put them on the surface and then you just dab it like this. But sometimes it's much better to just do so by air drying the slides. That way you don't physically separate the bacteria from your slide. So in this case, I check off the excess of iodide, and then again I put it sideways like this, and I start gently squirting water. So there's always a rinse in between the steps of the gram staining. And I shake off the excess of water, and then put the colorizing agent, 10 to 20 seconds. Then shake off the excess, another rinse. Shake the excess of water, then add the counter stain. 1 minute. Okay, so while I wait for this last part of the gram staining to go, so gram-positive bacteria stains purple and gram-negative stains red. So when you add your crystal violet, which is the primary stain, you introduce this into both gram-positive bacteria and gram-negative bacteria. So this dye stains the cytoplasm of both. And in the next step, when you are at the iodide, you create crystal violet iodide complexes that will get trapped in gram-positive bacteria when you add the decolorizing agent. But they will disappear, these crystal violet iodide complexes, from gram-negative bacteria, because gram-negative bacteria has less peptidoglycan in its cell wall. So when you add the decolorizing agent, which is alcohol, you create pores in this peptidoglycan layer, which is very small in the gram-negative, and through these pores, the crystal violet iodine complexes live. So basically, after the alcohol step in gram staining, gram-positive will always look purple, but gram-negatives will look colorless. So that's why you need to add your your secondary staining. So now I'm going to rinse, and then I'm going to keep splaining. So again, you just angulate a little bit your slide, and then make sure that you don't have any safranin. And then you just shake off the excess of water and let it to be vertical like that until they dry. And then when you add your counter stain, your gram-negative bacteria will stain with safranin, which is red. And this is the reason then why gram-negative stain red and gram-positive stain purple, because these gaps that you open with the decolorizing agent in the peptidoglycan from the from the gram-negatives, make this crystal violet iodine complex to escape And with alcohol, the decolorizing agent in gram-positives, you trap them because you seal actually the peptidoglycan layer as you are dehydrating with the alcohol the peptidoglycan. So once then these are dry, you just observe them under the microscope, and then you have to clean up your area afterwards. So this is all for this video, and I will see you in the next. Bye-bye.